Hi everyone, greetings to our lecturer Dr. Nur Ashikin and also my fellow groupmates. We are from Subgroup 1 and will be presenting the poster, and the title is Development of an Eco-Friendly De-Inking Process for the Production of Bioethanol Using Diverse Hazardous Paper Waste. Now, this title is researched by Sonu Saini, Preeti Chutani, Prabhat Kumar and also Krishna Kant Sharma. Now we shall move on to the introduction of this research title. Now, unlocking sustainable energy sources is imperative in combating environmental challenges and meeting escalating energy demands. Lignocellulosic bioethanol emerges as a promising solution, boasting advantages over fossil fuels such as reduced greenhouse gas emissions. Now, leveraging paper waste, including old newspapers and laser printed papers as feedstock for bioethanol production is not only a sustainable practice, but also essential for curbing pollution from the paper and pulp industry. However, hazardous ink in paper waste presents a significant obstacle to efficient bioethanol production, necessitating effective de-inking technologies. Enzymatic hydrolysis, particularly through the use of cellulases and xylenases, offers a preferred method over chemical approaches, demonstrating promise in breaking down complex polysaccharides into fermentable sugars. So, this study aims to compare enzymatic, ozone, and chemical de-inking methods for various paper samples, striving to identify the most effective approach for ink removal and to evaluate de-ink paper waste for bioethanol production, thus contributing to the advancement of renewable energy development.
I'll walk you through the methodology employed in this study. It has 9 phases. The first phase is microorganism and culture conditions. We begin by obtaining fungal culture, specifically Trichoderma citrinoviride MDU1, Aspergillus oryzae MDU4, and Ganoderma lucidum MDU7 were maintained on specific media at prescribed temperatures. Second phase is production of cellulase and lacase. Enzyme production was carried out via solid state fermentation using a wheat bran substrate with an appropriate inoculum. Cellulase activity CMCAs and FPAs and lacase were measured. Partial purification was achieved through ammonium sulfate salt precipitation and dialysis. Third phase is pulp preparation. Our pulp preparation involved optimizing the process using a laboratory-designed pulp maker. Various paper waste samples like EP, NP, and PP were shredded, soaked, and analyzed for sugar, phenolates, chromophores, and hydrophobic compounds. An incubation time of 2 hours was chosen based on consistent result. The fourth phase is dinking of paper samples. Dinking treatments included cellulase, lacase, ozone, and chemical methods with varying conditions tests for each treatment. For the next phase, which is structural and ultra-structural study of dinking papers, we utilize ATR, FTIR, SEM, TEM, and SRD to analyze surface functional groups, morphology, and cellulose crystallinity. Sixth phase, crystallinity measurement of dinking papers. Crystallinity of cellulose was assessed using X-ray diffraction SRD analysis. Samples included control, chemically dinked, and the most suitably dinked paper sample. The next phase is enzymatic hydrolysis of dinked papers. Optimally, dinked paper sample were subjected to enzymatic hydrolysis with saccharification yield calculated based on release reducing sugars. We move to the next phase, which is fermentation of sugar hydrolysates. Sugar hydrolysates were fermented using Saccharomyces cerevisiae and CIM3640, and bioethanol productions was assessed using chromic acid method. The last phase is analytical methods. The NSA reagent was used to analyze total reducing sugars. HPLC was employed for sugar analysis. The following Chocolcure reagent method was utilized for total phenolates estimation.
In the journey of the environmentally friendly and efficient process, the study embarked on the journey to optimize the pulp preparation by utilizing a curved wooden blade. High quality pulp was achieved in astonishingly short time frame of just 2 hours. This enhancement in productivity reduced the energy consumption, aligning with the commitment for a sustainable practice. Moving on to the crucial aspect of the inking methods, through meticulous experimentation, it was discovered that cellulose treatment effectively removed ink components from newspapers, while lacase treatment holds promise for photocopier paper. Additionally, ozone treatment emerged as the optimal choice for printer paper. These tailored methods ensure the efficient ink removal while preserving the quality and the structure of the paper, mitigating the yellowing and the byproduct formation often associated with chemical de-inking methods. Enzymatic treatments, particularly cellulase and lacase, exhibit a significant ink removal while preserving the paper quality and the structure better than their chemical counterparts. Structural and ultra-structural analysis further validated the efficiency of the enzymatic and the ozone treatments in breaking down the ink particles and altering the paper surface properties. The study delved into the crystallinity measurement and the enzymatic hydrolysis. Enzymatic hydrolysis of the ink papers yield a substantial amount of reducing sugars, with optimization strategies further enhancing the sugar yields. Through fermentation using the Saccharomyces cerevisiae, ethanol was produced with a higher efficiency compared to the previous studies on newspaper and office paper. Ethanol production peaked at 24 hours, underscoring the immense potential for bioethanol production from recycled paper.
In conclusion, study has demonstrated the effectiveness of utilizing innovative enzymatic and dinking processes for transforming paper waste into valuable resources. The first value is efficient waste utilization. By employing cellulase and lactase enzymes alongside ozonation and enzymatic dinking techniques, we've effectively degraded ink components in paper waste. The next value is sustainable bioethanol production. Through the synergistic action of an enzymatic consortium and an industrial yeast strain, we've successfully converted ink waste papers into bioethanol. This approach not only facilitates sustainable biofuel production, but also aligns with our environmental sustainability goals, promoting a circular economy and reducing reliance on fossil fuels.
This work is acknowledged by the Council of Scientific and Industrial Research, CSIR, Government of India, New Delhi.
That's all from us. Thank you.